Well, good morning, everybody, and welcome back to Metabolism 2. Hopefully, you've had a wonderful and relaxing reading week. Just a couple things we got to get through. Midterm grades were posted, as you can see. Um, Not this week, obviously, because I'm not here, but the following week, we will hold exam viewings. I'll post an online sort of like Excel type of sheet that everybody can register whatever time and date works for you, so everybody can have a chance to view their exam, and we can go through it together. Um, but yeah, so as you can obviously tell, this week we're going to be online and virtual, asynchronous. I'll try to upload the videos if everything works out during the regular like 8:30 a.m. time Monday, Wednesdays that were scheduled. But if there is a delay or something goes wrong with the internet, it might be posted a little bit later. But again, hopefully you're listening to this at 8:30 on Monday and on Wednesday. But once I'm back, not this week obviously, but the following week, we'll be back in person. I'll make a Brightspace announcement so that everybody's very clear that the following week will be in person. But just wanted to get that out of the way right now, so there was no confusion. So with that being said, we are starting a brand new chapter. So this is the next part of metabolism 2. We're starting the biosynthesis of nucleotides. And a lot of it actually interconnects or ties in together with the amino acid biosynthesis pathways that we were talking before, and a lot of the precursors and a lot of the components that we saw previously in the amino acid synthesis will come through, and we'll actually see how all this starts connecting together and how certain molecules, specifically like PRPP or specific amino acids such as glutamate or glutamine or aspartate, they come back, right? And we'll actually be able to trace those molecules out, see where they come from, and see how they integrate into our biosynthesis of nucleotides. So, without further ado, let's get started. All right, well, let's begin. Let me see if I can get a laser pointer up and about. Okay, awesome. Let's start with the biosynthesis of nucleotides. Nucleotides are precursors to DNA and RNA, and they have a bunch of important functions in all cells. Not only are they precursors to DNA and RNA, they're essential carriers of chemical energy. So, a role primarily of ATP, right? We know that ATP is like that main source in the body that provides us energy. We also have GTP, right? GTP is used a lot for protein synthesis and things like that. But more importantly, they're actually components of specific cofactors such as NAD, FAD. We also have adenosylmethionine, coenzyme A. Uh, we also have a bunch of activated biosynthetic intermediates such as UDP glucose and CDP diacetyl glycerol. We also have some secondary messengers, specifically like cellular secondary messengers, such as CAMP and CGMP, right? So CAMP and CGMP, which are commonly known. And what we want to kind of focus on really is that there's two pathways that leads to the production of nucleotides right here, right? So we have the de novo pathway and we also have the salvage pathway. Um, so the de novo synthesis or the de novo pathway of nucleotides begins with metabolic precursors such as amino acids, ribose 5-phosphate, CO2, and NH3. And that's kind of like the base of it of where it becomes, right? So you kind of think of de novo means new, so you're building it from brand new, from scratch, right? We also have the salvage pathways. So the salvage pathways recycle the free bases that are still around and nucleosides released from nucleic acid breakdown, right? So remember, there's a difference between nucleotides and nucleosides. Um, Both of these pathways are important in cellular metabolism, and we'll talk about them a lot more a little bit later on, and specifically a lot more in focus and detail. So, a little more um, getting into it. So, the de novo pathways for purine and pyrimidine biosynthesis seem to be nearly identical in all living organisms. So, whether it be all eukaryotes, all mammals, basically, they're the same everywhere. Um, so notably, the free bases guanine, adenine, thymine, cytidine, and uracil are not intermediates in these pathways. So that is that the bases are not synthesized and then attached to ribose, as you would think. The purine ring structure is actually built up one or a few atoms at a time attached to ribose throughout the process. So pyrimidine ring is synthesized as orotate, and we'll see that a little bit later. We'll go through these pathways a lot more in detail later um, in the slide deck. But anyways, as it's synthesized as orotate, then attached to the ribose phosphate, and then converted to the common pyrimidine nucleotides required in nucleic acid synthesis. Um, Although these free bases are not intermediates in the de novo pathways, they are intermediates in some of the salvage pathways. So we keep that in mind too. But we also have a bunch of other important precursors that are shared by the de novo pathways for synthesis of pyrimidines and purines, specifically our good old friend PRPP that we all have come to love um, and know very well. So PRPP is important in both of these pathways. And in these pathways, the structure of ribose is retained in the product nucleotide. So in contrast to how in the tryptophan and histidine biosynthetic pathways, um, that the structure isn't retained. Now it will be retained. And we'll see that a lot again in the later pathways. So, an amino acid is an important precursor in each type of pathway. So, glycine is an important amino acid for purines, and aspartate is the important amino acid for pyrimidines. Glutamine, again, is the most important source of amino groups as we've seen many, many times. Again, that's why we went through all those examples with glutamine in the previous chapter, the amino acid chapter. Um, it's like I said, it's the most important source of amino groups. So, in 5 different steps in the de novo pathways, we'll actually see where glutamine actually is the source of amino groups. Um, we also have aspartate over here, is also used as the source of an amino group in the purine pathways in 2 steps. Um, Two other things actually deserve some mention that we should probably talk about. First, there is evidence, especially in the de novo purine pathway, that the enzymes are present as large multi enzyme complexes in the cell. Something that's a reoccurring theme in our discussion of metabolism, right? That these large multi complexes come together working with individual subunits, right? And we'll see that a little bit later on, but we'll have alpha 2, beta 2 subunits. Some of them have alpha 4, beta 4. Some of them even have alpha 4 and beta 6 subunits. So they come together with one goal, um, and it's a recurring theme in this in metabolism. Second, something that we should keep in mind, and it's quite interesting, is that the cellular pools of nucleotides, other than ATP, this is very important, are quite small. Usually around less than 1% is available readily. So again, ATP is always available, but the other nucleotides are quite small. Like I said, perhaps 1% or less of the amounts required to synthesize the cell's DNA. So if you have less than 1% of what's required to synthesize DNA, cells must continue to synthesize nucleotides during nucleic acid synthesis. And in some cases, nucleotide synthesis may limit the rates of DNA replication and transcription. So, because of this importance that it's constantly being made and that we have a constant demand for it, um, it's actually become quite the important target for medicine. We'll see a little bit later that it's actually quite a big target for a lot of cancer therapy and a lot of other disease and drug treatments. So over here we can actually see de novo purines, and we'll just kind of go over it a bit. So paranucleotides of nucleic acids are adenosine 5 monophosphate, so AMP, and guanosine 5 monophosphate, so GMP. So like I said, the two parent purine nucleotides are AMP and GMP, containing the purine base adenine and guanine. But what I want to highlight over here is just this figure over here, because we didn't really go through it as much in the other slide as we should have. So right here, we kind of show the origins of the carbons and nitrogen atoms of the purine ring system. So you can see over here that we have a couple nitrogens, amide N of glutamines that are being donated right here. We have this carbon over here donated by formate. We also have this structure over here donated by glycine. This single carbon over here donated by CO2, single carbon over here donated by formate, and this single nitrogen over here donated by aspartate. And it'd be pretty good if you guys knew this and understood this and knew, you know, where this would come from if it were to be on a midterm or something like that. But what's pretty cool about this is this was actually determined back in the 1950s by a gentleman named John M. Bushnan. And what he did was he used isotopic tracer experiments in birds. So this was all done using birds in the 1950s, and they had radio labeled isotopes. And that part isn't too important how the isotopes work. But what's cool is that this actually happened over 70 years ago, and we understood where each component comes from. All right, well, I guess we're going through this nice, scary pathway, but we'll break it down step by step, and hopefully, we can make it easier to understand, and you guys can grasp the concepts quite easily. So, in the first committed step of the pathway, right here, we have in the amino group, which is donated by glutamine. As you can see right here, glutamine becomes glutamate. You also release PPI. We're going to attach it to our wonderful good old friend PRPP at the carbon 1 of PRPP. So right over here, you can see that we have the attachment of our nitrogen group um, from glutamine. So the resulting molecule, which is this 5-phospho beta D ribosylamine, um, this molecule right here, highly, highly unstable, has a half-life of 30 seconds, maybe even less, at physiological pH, which is roughly, 7.3 to like 7.6 pH. So from here, the purine ring is subsequently built upon this structure right here. So this is the foundation of the ring. So the pathway described here is identical in all organisms, like I said before, with the exception of actually one step, which is step 6, that differs in higher eukaryotes. But we'll get that on the other slide a little bit later when we actually hit that part of the pathway. The second step over here is the addition of 3 atoms from glycine. So we're going to take our wonderful friend glycine, donate 3 atoms. You also have ATP, so you need to input energy to actually get this going. So again, you're going to release ADP plus a PI over there. So the ATP is consumed to activate the glycine carboxyl group in the form of an acyl phosphate. And this right here is, well, a condensation reaction. So the added glycine amino group is then formulated by um, N10 formal tetrahydrofolate 3. I know that's a mouthful, but that is the next step. And just so there's no confusion, over here, this part right here, this R group represents the rest of the PRPP molecule. So this nitrogen right here, this green nitrogen, is still this green nitrogen over here that's attached to this unstable molecule over here with PRPP base. Um, and then a nitrogen is contributed by glutamine in step 4. So we have over here, we have our wonderful friend glutamine again. And then we have, again, you have to input ATP, you get your ADP plus PI reaction. Um, and then right before anything else happens, you also have, um, before dehydration and ring closure yield, the 5-membered imidazyl ring of the purine nucleus becomes AIR over here. And what we want to take away from this over here, right, is just kind of understanding what amino acid is donating um, what component um, to each part of the structure, right? So you want to know the glutamine is donating the nitrogen to that carbon 1 at PRPP. You want to know that glycine over here is donating this 3 carbons on the branch molecule. Something that's important too that we want to highlight over here is that the transition from this molecule over here, FGAM, to AIR, in the input of ATP, closes the ring structure and gives us that imidazole group um, that um, we're constantly seeing over and over again. And something else, for this one, we don't really want to know or care too much about the enzymes that catalyze these reactions. This isn't really important. Um, Again, knowing sort of the structures here, and knowing um, the short form names for everything will suffice, right? So PRPP is fine here. This intermediate right here, you should probably know 5-phospho-beta D ribozolamine. You can know this as GAR, FGAR, FGAM, AIR. That's perfectly fine for nomenclature. You just kind of want to understand what's happening right here. So at this point, 3 of the 6 atoms needed for the second ring in the purine structure are in place. And so to complete this process, we need to go to the next slide to see what's happening. All right, so like we said a little bit earlier, um, at this point, 3 of the 6 atoms needed for the second ring in the purine structure are in place. So to complete the process, our a carboxyl group, sorry, is first added, as you can see here in step 6. This carboxylation is unusual in that it does not require biotin, but instead uses the bicarbonate usually present in aqueous solutions. So the fact that this bicarbonate is actually here in this reaction instead of biotin is actually quite odd and something that we should take note of. A rearrangement transfers the carboxylate from the exocyclic amino group to position 4 of the imidazole ring over here in step 7. Um, so step 6 and step 7 are only found in bacteria and fungi. In higher eukaryotes, including humans, um, the 5 amino imidazole ribonucleotide product of step 5 from the previous slide, so AIR, is carboxylated directly to carboxyamino imidazole, so CAIR over here, right? So in humans, we get to skip step 6 and step 7, and we can just use this 6A process over here and end up here instead. And since it is an important step, um, we do want to know what enzyme actually performs this. So step 6A, you want to know that it's AIR carboxylase is the enzyme that catalyzes the reaction from AIR to CAIR. From now, we want to notice that aspartate is the next amino acid that comes in and donates its amino group in 2 steps, in both steps 8 and 9. So as you can see right here, step 8, we have aspartate coming in. Again, we have that input of ATP, release of ADP plus PI. You can see that this skeleton right here by aspartate is donated. We end up with this molecule SAICAR, or I don't know how to pronounce it, sidecar. That's how I would say it. Um, and from here, step 9, you have a release of fumarate right here. And then, ooh, my little laser's falling off the screen. Pumps or jumps over here to this molecule over here where my laser is now at, which is AICAR. And what's happening here, right, is that formation of an amide bond followed by elimination of the carbon skeleton of aspartate, and you're releasing it as fumarate, right? So as you can see right here, when fumarate leaves, this carbon skeleton also leaves, and the only thing that's left in this molecule over here is this nitrogen over here. And lastly, the final carbon is contributed by our N10 formyl tetrahydrofolate over here in steps 10. And the second ring closure takes place to yield the second fused ring of the purine nucleus, which is step 11. In step 11, you have that release of H2O, so your water, and that gets us to our final molecule over here, which is this IMP molecule. And here we can see the formation of IMP, where IMP is that first intermediate with a complete purine ring that's completely closed. And as you can see right here, we have all the carbons, all the nitrogens donated from different groups, right? So we have that CO2 donation from here, whereas in bacteria and fungi would actually be donated by the bicarbonate in the aqueous solution. We have our carbon-carbon nitrogen over here donated by our amino acid glycine. We have nitrogen group over here donated from our aspartate. where it had that original carbon backbone, but that carbon backbone leaves as the fumarate over here. We have our donation of our last carbon by the formal group over here by our N10 formal H4 folate, right here and right. And then we also have donations from our nitrogens over here from our other groups, such as glutamine that was previously donated as well. So as you can see right here, many components coming into this intermediate IMP, with the foundation, again, that base right here coming from PRPP, that we've all known. And from here, IMP continues on, and we'll actually see the biosynthesis of AMP and GMP from this intermediate IMP. So here things start coming together where we can see um, our friend IMP actually creating more useful products, such as AMP and GMP that we're a little bit more familiar with. So the conversion of IMP to adenylate requires an insertion of an amino group derived from aspartate. So this process right here takes place in two reactions similar to those, to introduce, as used to introduce the N1 of the purine ring in steps 8 and 9, as we previously saw before. However, though, a crucial difference between this is that it is GTP that is used rather than ATP. So as we can see over here, we have IMP. We start off, we have aspartate donating some sort of, carbon skeletal backbone with that nitrogen right here. We have GTP coming in, donating it, and again you have GDP plus PI leaving, and you end up with this intermediate right here, which is your adenylosuccessinate, and then again, just like before, we have fumarate that leaves. So it leaves in that carbon skeleton backbone, and you end up with just having that nitrogen group right there. So like I said, it donates that amino group in two steps, as previously before, and we have that elimination of the carbon skeleton of aspartate, leaving as fumarate, and we end up with, well, AMP, the molecule that we actually need for this process. On the other hand, we actually have the formation of guanylate, so GMP. So guanylate is formed by the NAD plus, requiring oxidation of IMP at carbon 2. So we can see it right here. We have water coming in, we have NAD plus, and then we have NADH plus H plus leaving. You can see the water oxygen over here is integrated over here into that carbon 2, followed by the addition of an amino group derived from glutamate. So we have our intermediate over here, which is XMP, or xanthylate. It comes in, you have your amino group derived from glutamate. Um, you have your input in this case, which is very important, which differs from AMP. We have GTP over here, and then over here we have our energy source coming from ATP. You have your release of AMP plus your PPPI, and then you end up with your molecule over here, guanylate GMP. Um, and that is the final step. And now we have both AMP and GMP from our IMP. So a common theme that's been reoccurring over again is this regulation, right? We saw the regulation of amino acids, and now we need to regulate these these purines, right? That tight regulation. We need them at a specific time. We need them at a specific quantity. Sometimes we need less. Sometimes we need more. So we need to tightly regulate these processes over again. So there's three major feedback mechanisms, mechanisms cooperate in regulating the overall rate of de novo purine nucleotide synthesis. Um, and the relative rates, they also regulate the relative rates of formation of the two end products, adenylate and guanylate, that we saw previously, that came from IMP, right? So we have IMP, and then we have our final products, which are GMP and AMP. So the first mechanism is exerted on the first reaction that is unique to purine synthesis. So it's at the transfer of an amino group to PRPP to form that intermediate, that 5-phosphoribosylamine. So right here we have our PRPP to that molecule right here, that intermediate. That intermediate right there is the one that I talked about a little bit earlier and said that it was highly unstable, only lasts about 30 seconds at physiological pH, right? So this right here is one of the most or the most important regulation mechanisms that we have. So this reaction right here is catalyzed by the allosteric enzyme glutamine PRPP aminotransferase, which is inhibited by the end products IMP, GMP, um, and AMP. So you can see right here we have IMP, GMP, and AMP, which inhibit this enzyme right here. So therefore, whenever we have AMP or GMT accumulation, when it becomes excess, the first step in the biosynthesis um, from PRPP is partially inhibited, right? So we get this partial inhibition right here. In the second control mechanism, exerted a little bit later, an excess amounts of GMP right here in the cell inhibits formation of xanthylate. And we remember xanthylate is one of the intermediates from IMP to GMP. So it inhibits the formation of xanthylate from IMP by IMP dehydrogenase. So you can see right here we have IMP dehydrogenase. Without affecting the formation of AMP, right? Because a lot of times, just because we don't need one molecule doesn't mean we need another molecule, even though, you know, high levels of GMP would affect or inhibit the formation of this intermediate right here, 5-phosphorybosylamine. But like I said a little bit earlier, it only partially affects this regulation right here. So you would still get production of AMP. Um, Conversely, though, an accumulation of adenylate inhibits the formation of adenylosuccinate um, by adenylsuccinate synthetase. Oh my, hard to say a lot of these ones. So right here, this enzyme right here, without affecting the biosynthesis of GMP. So again, if we have high levels of AMP, we can self-regulate and cut off um, the formation of AMP without affecting the levels of GMP. However, though, when both products are present in sufficient quantities, IMP builds up and it inhibits an earlier step in the pathway. This regulatory strategy is called sequential feedback inhibition, which we've seen previously in our amino acid synthesis pathways. So, in the third mechanism, GTP is required in the conversion of IMP to AMP, if we remember that correctly. Whereas ATP is required for conversion of IMP to GMP. So that's just the slide before. A reciprocal arrangement that tends to balance the synthesis of the two ribonucleotides. And then the final control mechanism is the inhibition of PRPP synthesis by the allosteric regulation of ribose phosphate pyrophosphokinase. So this enzyme is inhibited by ADP and GDP, in addition to metabolites from other pathways for which PRPP is a starting molecule. So for this slide, what we want to take away from this, right, is that, we have that tight regulation of this purine, of these purines, right? AMP and GMP, we can regulate them in one, two, three, and even potentially like a fourth way, right? One that may not be included in this diagram. And just goes to show the levels of regulation and the levels that the body goes to carefully regulate all of these biomolecules that are constantly needed. And from here, we should probably know, what molecule inhibits what enzyme. We should know the names of these molecules. We should know AMP, GMP, IMP, right? They regulate this enzyme. AMP can regulate this enzyme. We should know that this right here is a type of sequential um, inhibition, right? Um, and we should understand all those concepts here. And we should understand all these names here from this slide over here. Yeah. All right, shifting gears now. So now that we looked at purine biosynthesis, it's now time to look at pyrimidine biosynthesis over here. So the common pyrimidine ribonucleotides are cytidine 5 monophosphate, so CMP, and uridine 5 monophosphate, UMP, which contain the pyrimidines cytosine and uracil. Um, yeah. So anyway, so de novo pyrimidine nucleotide biosynthesis proceeds in a somewhat different manner from purine nucleotide synthesis. So this right here is kind of the pathway of it, and we'll look into it just a little bit in a second. But it is a little bit different, and the main thing why it's different is that the six-membered pyrimidine ring is made first, then it is attached to the ribose 5 phosphate. So you can see right here just very quickly that this pathway starts off, you have some sort of product, then it's fused the ring continues to have some modifications to it, and then from here it's finally fused to that ribose 5 phosphate. Whereas before, everything was kind of built together right from that foundation of PRPP. Um, so one thing we also want to note right here is that we have in this reaction is that we require carbamyl phosphate right here, which is also an intermediate in the urea cycle. Um, and in animals, the carbamyl phosphate is required in urea synthesis, is made in the mitochondria by carbamyl phosphate synthetase 1. However, though, the carbonyl phosphate required in pyrimidine biosynthesis is made in the cytosol by a different form of the enzyme, which is carbonyl phosphate synthetase 2. And that's a key thing we really, we get to highlight again. I know back in the earlier, lectures that we had, we talked about how the body tends to regulate a bunch of processes and it tends to use different mechanisms, right? It can change physiological pH, it can change um, the quantities of a product. But one of the things here that we highlighted before is that it can actually change the location of where things are made. So here we can actually see the carbonyl phosphate is sometimes made in the cytosol, whereas in now, um, pardon me, before it was made in the mitochondria, and now for pyrimidine biosynthesis, carbonyl phosphate is made in the cytosol. Um, by a different form of the enzyme. However, though still the same enzyme, but a different form. And by changing the location of where this enzyme is present, whether it be the mitochondria, whether it be the cytosol, you can actually further regulate these molecules and further regulate um, these processes even more. So in bacteria, a single enzyme supplies this carbonyl phosphate right here for the synthesis of arginine and pyrimidines. And this bacterial enzyme has three separate active sites spaced along a channel nearly 100 angstroms in length. And bacterial carbonyl phosphate synthesis provides um, a cool illustration of something we'll touch upon a little bit later, which is that in these multi-enzyme complexes form together, but we'll get in that a little bit later. But anyways, to start right now, to start this pathway. So first we have carbonyl phosphate over here, and it reacts with aspartate right up here to yield N-carbonyl aspartate. So this is our first molecule right here, N-carbonyl aspartate. You can see it right here. It's not a closed ring yet, so it's just this carbon structure right here. However, though, this is the first committed step of pyrimidine biosynthesis. So the moment you have started this pathway and you have this first molecule, the body will commit to the biosynthesis of pyrimidines. So this reaction is catalyzed by aspartate transcarbamylase. Um, Aspartate transcarbamylase right here, as you can see. Um, and in bacteria, this step is highly regulated. And bacterial aspartate transcarbamylase is one of the most thoroughly studied allosteric enzymes known to mankind. Okay, but moving forward, the removal of water um, from N-carbonyl aspartate. Um, as you can see right here, we have the removal of water from our molecule by our enzyme dihydrourotase. the pyrimidine ring is closed to form L-dihydrourethate. So you can see right here, we have the removal of water. You can see now that the ring or the previous structure is now closed to form this nice ring structure. And now we have our molecule L-dihydrourethate. So this compound is then oxidized to the pyrimidine derivative orotate. So you can see right here we have orotate, we have NAD plus coming in, and we leave with our NADH plus H plus. So this compound is oxidized, a reaction in which NAD is the ultimate electron acceptor. Okay, so it's NAD that is the ultimate electron acceptor. In eukaryotes, the first three enzymes in this pathway, um, which happen to be carbonyl phosphate synthetase 2, which remember was in the cytosol, not in the mitochondria as carbonyl phosphate synthetase 1. Um, so carbonyl phosphate synthetase 2, aspartate transcarbamylase, and dihydrourethase, so enzyme 1, enzyme 2, and then the previous enzyme. So those three enzymes together um, are part of a single trifunctional protein. So this protein, known by the acronym CAD or CAD, contains three identical polypeptide chains, each one, each chain about 230,000 in molecular weight. Um, and each of these have active sites for all three reactions. So this kind of just hints at that concept that we've, touched base upon a few times, is that we have these large multi-enzyme complexes, coming together to form, perfect conditions, perfect, scenarios where these reactions can happen together. I mean, a lot of the times, the reason why they're so clumped together happens to be because, one, if you're close by one another and you have an unstable intermediate, quickly, if you're close by, that unstable intermediate can actually go over to the next active site, and, the proper modifications to make it, highly unstable to stable can be quickly done. And two, efficiency processes, right? The body hates wasting energy. It hates being, wasteful. So if everything can be done together in a single place, with this, the same conditions, again, the body loves that. And it's something that we've seen over and over again um, come up, um, that theme. Okay, so moving forward. So once orotate is formed, we have our wonderful molecule here, orotate now. The ribose 5 phosphate side chain is attached to yield orotidylate. Oh my, I can't say it. But what we want to know is that this ribose 5 phosphate side chain is provided by, once again, our wonderful friend PRPP. So PRPP comes in again, donates that ribose 5 phosphate side chain, and we end up yielding our wonderful molecule over here, orotidylate. So from here, orotidylate is then decarboxylated to UMP. So we have that removal of that CO2 group right here, and you form our UMP. From here, UMP is phosphorylated to UTP. So you can see right here, we have the addition of two ATPs, the removal of, well, the release of two ADPs, and we form our good old friend UTP. Um, so from here, it gets a little bit interesting. So from here, we can go from UTP to CTP. And what we want to take note here is that CTP is formed from UTP by the action of citylate synthetase. So citylate synthetase right here, by the way of an acyl phosphate intermediate. And what that does is that it consumes one ATP to get us our CTP. So from here, the nitrogen donor is normally glutamine. And again, if we have glutamine here, we're going to end up using glutamate over here. Although, and this is a sidebar, the citylate synthetase, So this enzyme right here, in many species, can actually just use NH4 directly, and it can completely skip the use of glutamine. So that's also something to keep in mind. Okay, so shifting gears a little bit over here, we've been focusing a lot on pyrimidine synthesis, but now we got to focus on pyrimidine nucleotide regulation. So regulation of pyrimidine nucleotide synthesis occurs through aspartate transcarbamylase, or ATCase, or ATCase, or however you want to say it. Um, so in bacteria, this regulation occurs in large part through, like I said, ATKs, or aspartate transcarbamylase, however you want to think about it, which catalyzes the first reaction in the sequence, and it is inhibited by CTP. So CTP, again, if you remember, is our final end product um, of the sequence. So the end sequence ends up regulating that first um, um, enzyme, so aspartate transcarbamylase. Um, so the bacterial ATCase molecule consists of 6 catalytic subunits and 6 regulatory subunits, which we'll see a little bit later. Won't be too important looking at the structure. What's more important here is understanding how this kind of gets regulated. Um, so the catalytic subunits bind the substrate molecules, and the allosteric subunits bind the allosteric inhibitor, which is CTP. So the entire ATCase molecule, as well as its subunits, exists in 2 conformations, active and inactive, which is what makes the most sense. When CTP is not bound to the regulatory subunits, the enzyme is maximally active. So as you can see over here, um, the enzyme is maximally active when CTP is not bound to the um, regulatory subunits. As CTP accumulates, though, and binds to the regulatory subunits, they undergo a change in conformation. So as you can see right here, if you start adding CTP, um, eventually you start reducing the activity of this enzyme right here. Um, and this change um, of this enzyme, so a change, you can prevent the change in this enzyme actually by binding ATP, which is pretty interesting. So if we look at this figure a little bit more, when we have high levels of CTP, so we have high levels of our final product over here, CTP, we have a reduction obviously in, you know, the activity of this enzyme right here. When we have no CTP and we have normal activity, we have, regular speed. However, though, when you have CTP plus ATP, you actually regain this function by quite a bit, actually. It's not the same levels to which it was before with no CTP. However, though, you can see right here that addition of ATP prevents changes induced by CTP. And that's one of the key things you want to take away from this, right? Um, is that as you increase your concentration of aspartate right here along this right here, and you have an increase in CTP, you're going to get a decline in activity of this enzyme. But what's really important is that you want to highlight is that ATP can prevent the changes induced by CTP. Again, not to the normal activity, but quite close to what it was before. All right, so shifting gears here a little bit, we're going to go to nucleoside monophosphates and their conversion to nucleoside triphosphates. So, nucleotides, to be used in biosynthesis, are generally converted to nucleoside triphosphates. The conversion pathways are common to all cells, like we've said before. Um, this is a common theme that keeps reoccurring. So, phosphorylation of AMP to ADP right here is promoted or done through adenylate kinase, in the reaction, ATP plus AMP gets you 2 ADP. So you can see right here AMP to ADP, and this is a reversible reaction, so keep that in mind. The ADP formed is phosphorylated to ATP by the uh, glycolytic enzymes or through oxidative phosphorylation. So ATP also brings about the formation of other nucleoside diphosphates by the action of a class of enzymes called nucleoside monophosphate um, kinases. And these enzymes, which are generally specific for a particular base, but not but non-specific for the sugar, so the ribose or the deoxyribose, catalyze the reaction ATP plus NMP um, in a reversible reaction gets you ADP plus NDP. Um, the efficient cellular systems for rephosphorylating ADP to ATP tend to pull this reaction in the direction of the product. So we usually end up with ADP plus NDP. We also have nucleoside diphosphates are converted to triphosphates by the action of a ubiquitous enzyme called nucleoside diphosphate kinase, which catalyzes the reaction NTPD plus NDPA gets you NDPD plus NTPA. And this enzyme is notable in that it is not specific for the bases, so the purines or the pyrimidines, or the sugar, which is the ribose or the deoxyribose. So this non-specificity applies to both phosphate acceptor and the donor, where the acceptor is the A, you can see over here, and the D is the donor over here. Although the donor, which is NTPD, is almost invariably ATP because it is present in higher concentration than other nucleoside triphosphates under aerobic conditions. So we said that a little bit earlier before too, right? We said that ATP is generally in high concentrations, and other molecules such as UMP, CTP, whatever, are well beneath that 1% threshold. So we continuously need to be making them. So in this case, ATP is always available. So ATP is almost always this NTPD donor over here that you're seeing. So kind of continuing on before with that whole um, um, prospect of nucleotides, ribonucleotides, we're kind of going into now that ribonucleotides are the precursors of deoxyribonucleotides. So, deoxyribonucleotides, which are the building blocks of DNA, are derived from the corresponding ribonucleotides by the direct reduction of the secondary carbon atom of the D ribose to form the 2-deoxy um, derivative. So, for example, we have something like ADP is reduced to 2 prime deoxy adenosine phosphate. So D ADP and so on, like GDP is reduced to D GDP, right? Um, so this reaction is actually somewhat unusual in that the reduction occurs at a non-activated carbon, um, which is there's no other chemical reactions that are actually like this. And this reaction right here is catalyzed by this ribonucleotide reductase, which is this image over here. And we'll go into great detail and learn about it in the next few slides and how it actually works. And we'll know all about the substrates. We'll learn about the regulatory sites, the primary regulatory sites, the beta unit, the alpha units, and so on and so forth. So we'll spend a lot of time on this molecule here. So the reduction of the D-ribose portion of a ribonucleoside diphosphate to the two primary deoxy D-ribose requires a pair of hydrogen atoms, which are ultimately donated by NADPH via the intermediate hydrogen carrying protein thioredoxin. And we'll see that a little bit later and how that plays. So this ubiquitous protein serves a similar redox function as seen in photosynthesis and other processes. So what thioredoxin does is it has a pairs of thiol groups, so it has SH groups that carry hydrogen atoms from NADPH to the ribonucleoside diphosphate. So you can see right here you have those SH groups right here, and you're going to eventually carry them to the ribonucleoside diphosphate. And a little bit later, we'll actually go through the mechanism very, very carefully, and we'll see how this bridge actually works and forms. Um, so from here, its oxidized form, which is the disulfide, because this ends up forming a disulfide bridge right here, is reduced by NADPH in a reaction catalyzed by thioredoxin reductase, and reduced thioredoxin is then used by ribonucleotide reductase to reduce the nucleoside diphosphates. Two, you get your deoxyribonucleoside diphosphates. So you go from your NDPs to your DNDPs. From here, we also have a secondary form source of reducing equivalents for ribonucleotide reductase, which ends up being glutathione, so GSH, which we learned a little bit before in the amino acid section. So glutathione serves as the reductant for a protein closely related to thioredoxin, which is called glutaredoxin, which is then transfers the reducing power to the ribonucleotide reductase. So this protein, this enzyme, this ribonucleotide reductase is notable in that its reaction mechanism provides the best characterized example of the involvement of free radicals in biochemical transformations. Um, Once thought to be like extremely rare in biological systems. Um, so this exists in a alpha 2 beta 2 dimer with the catalytic subunits, these A2 alpha 2 units. Um, so these are the catalytic subunits, and the radical generation subunits beta 2 are down here. Each catalytic subunit contains two kinds of regulatory sites. So these alpha subunits right here have two different kinds of regulatory sites. Um, Here we have two active sites of the enzyme are formed at the interface between the catalytic, so that alpha subunit, and the radical generation beta subunits. So that right there is where the two active sites of the enzymes are formed. So you can see right here is the active site right here. We have this alpha subunit over here. We have the beta subunit right here. So in between these two subunits, we have the active site of this enzyme. At each active site, an alpha subunit contributes two sulfhydryl groups. So we have these two sulfhydryl groups right here, as you can see, um, required for activity, and the beta 2 subunits contribute a stable tyrosol radical. So over here, you end up having this tyrosol radical, as we'll see. So the beta subunits also have a binuclear iron. So you have this F3 plus complex, so F3 plus, F3 plus, and as you can see here, it's in the other subunit as well. And what this binuclear iron does is a cofactor that helps generate and stabilize tyrosine 122 radical, which we'll see a little bit later. But you can see it right here highlighted in this orange color right here. This is that tyrosine 122 radical right here. And we'll kind of see the importance of it a little bit later. And we'll go into much, much detail of this actual mechanism and how this actually works. Um, However, though, the tyrosol radical is too far from the active site to interact directly with the site. But we have several aromatic residues which form a long range radical transfer pathway to the active site. And I believe in my next slide we'll actually be able to see that. Um, and this right here is a likely mechanism for the ribonucleotide reductase reaction, um, as we'll see in the next slide. So the regulation of this ribonucleotide reductase is unusual in that not only its activity, but its substrate specificity is regulated by binding um, of effector molecules. So each alpha subunit has two types of regulatory sites. So you can see right here, we actually have multiple regulatory sites. One type affects overall enzyme activity and binds either ATP, which activates the enzyme, or DATP, which inactivates the enzyme. Um, the second type alters substrate specificity in response to the effector molecule. So we have ATP, we have DATP, DTTP, and DGTP. So as you can see right over here, we have that substrate specificity site. Um, and that's what's bound there. So when ATP or DATP is bound, um, reduction of UDP and CDP is favored. And I think we'll go to another slide to actually explain this a little bit better. Okay, so I think this slide does a better job of actually explaining what I wanted to explain. So I'll kind of just go over that again. So we have one location that actually affects the overall enzyme activity and binds ATP, which activates the enzyme, or DATP, which inactivates it. The second type, like I said, alters the substrate specificity in response to the effector molecule, whether it be ATP, DATP, DTTP, or DGTP that is bound there. So as you can see right here in this figure right here, when ATP or DATP is bound, we end up having a reduction um, of UDP and CDP when those two effector molecules are bound. When DTTP or DGDP is bound, we end up having a reduction in GDP or ADP, respectively. Um, so this scheme is designed to provide a balance pool of precursors for DNA synthesis. ATP is also a general activator for biosynthesis and ribonucleotide reduction. Um, and the thing here is the presence of the DATP in small amounts increases the reduction of pyrimidine nucleotides. And an oversupply of the pyrimidine DNTPs is signaled by high levels of DDTP. So your molecule over here, DTTP, which shifts the specificity to favor reduction of GDP. And if you have high levels of if you have high levels of DGTP, in turn, this shifts the specificity to ADP reduction. And we end up having these high levels of DADP shut the enzyme down. And these effectors are thought to induce, several distinct enzyme conformations with altered specifications. And we'll see a little bit later, actually, how we can actually change and alter the structure of this enzyme, which ends up actually changing and affecting its its overall um, productivity. So I think we'll end here. We'll just go through this pathway right here, and then I think this is a natural place for us to kind of end, and then we can continue on in the next lecture, the other parts. So this right here is a likely mechanism for the ribonucleotide reductase reaction um, that happens. So this right here is the proposed mechanism of ribonucleotide reductase that happens in eukaryotes, and so on and so forth. So right here, you just want to know, you want to just look at the fact that we have these activated thiol groups that are present on the alpha subunit. You also want to make note that this X right here is the active site radical that is on the beta subunit. Um, and that's kind of about it, that you kind of want to know. Um, and this X sometimes, not always, but sometimes in specific, depends on what organism you have. It is a thiol radical of cysteine 439. But anyways, so we start off over here with our NDP molecule, as you can see right here. And we have our active site radical right here, which ends up attacking the 3' ribonucleotide, and you end up forming this radical. So we end up forming this radical over here on this 3' um, of our NDP. So from here, what ends up happening is we have the 2' hydroxyl is protonated. So you can see right here from this one self hydrolyl group right here, we have the protonation of this group right here, and we go down here, and we can now see that we've now formed um, the protonation complete over here. Remember, we also have this free radical over here. This free radical came here. We have that hydrogen group that ended up binding to this active radical site over here. What ends up happening now is that we have H2O, so our water is eliminated to form a radical stabilized carbocation. So down here, we have this nice little carbocation um, that is stabilized. This radical is stabilized through this carbocation over here. From here, what we end up happening is that one of the thiols is oxidized on the enzyme, and the 2 electrons are transferred to the secondary carbon. So you can see now the secondary carbon over here now has the hydrogen over here, and now we've formed this disulfide bridge over here. Um, that's now happening. What ends up happening now is that the step one is reversed. Um, so you can see step one is reversed right here. So this radical is now going back, regenerating a tyrosine radical on the enzyme. So now we formed a tyrosine radical on the enzyme. We have now formed um, we have now brought back our hydrogen group back to our position 3 um, carbon on our original molecule. But now, because of this, we now have a DNDP molecule. And then step 6, ultimately to regenerate this disulfide group over here, the enzyme dithiol is reduced to complete the cycle. So you can see right here, we have a leaving of your DNDP, and you have your NDP coming back in. And this right here ends up converting um, our reaction from this disulfide group back into these individual um, thiols over here. And that's how we can go quickly from an NDP to a DNDP. And pretty much we'll leave it here for now. I think this is a good place to end the lecture. Um, on Wednesday, I will continue talking more about um, um, this whole process. We'll talk a little bit more about the degradation of purines and pyrimidines. And then from here, we will actually go into and talk a little bit more about how we can use these nucleotides and all these molecules we've just generated to actually target, diseases, cancer, gout, and so on and so forth. So the application part's coming up next. And then on Wednesday, we should tie this whole unit up. The nucleotide section is really short. I think Wednesday we should actually be able to finish about halfway through the lecture, and then we should be able to start the next section, which I believe we're going to do lipid biosynthesis next. Um, or, yeah, lipid biosynthesis next after this one, and then we'll continue on after that. Anyways, everybody have a good rest of your day. Uh, and then I will see you all virtually on Wednesday again for the rest of this lecture. Have a good one. Bye.